Hello, viewers, and welcome to our lecture series on biological techniques. Today's lecture will be on the techniques in ichthyofauna studies. Ichthyofauna studies basically refers to the study of fish in an area. And what are ichthyofauna? Basically, the ichthyofauna of an area basically refers to the composition of fish in an area, or the population or the different species of fish in an area is what ichthyofauna is. It refers to the composition and diversity of fish species in a given area. The ichthyofauna study is a very unique aspect of biology because the fish composition of an area, it's actually very vital because fish are a very important source of food and income. So we want to look at how these fish samples can be collected. We'll look at various techniques that are used for the collection of fish samples. The objective of any inventory is to identify all fish species in all habitats using all possible combination of gear types. Use different types of gear types to collect fish so that we are able to identify all the fish species in an area. For example, if we want to study the fauna of a river, let's say the River Non estuary or the River Non, there are different species of fish. So to collect fish that are representative of the entire area, or that could represent all the different species of fish, there is need to use different gear types because the species of fish are selective to certain gear types. There's gear bias. There's need to use different types of gear types so that the different species can be collected as representatives of the fish species in that area. A good study of biology of any fish species usually starts with a sampling procedure that is comprehensive enough to obtain samples that can be truly representative of the population under study. Okay, so the sampling technique is very important, the sampling procedure, so that it will be able to collect comprehensive data that's representative of the area under study. So various gear types are used for this study. To reduce the tendency of obtaining a biased sample, it is necessary to do the following. One, use more than 2 sampling gears to obtain samples. Then the second, use both passive and active sampling techniques. It is expected that both passive and active techniques are used to collect fish samples. The passive techniques include the use of traps, the use of long set lines, and the use of some nets such as gill nets and many other techniques. Then you can also use the active techniques such as the use of cast net, Then we can also use trawls or trawlers or seines, pole seine nets and beach seine nets can be used to collect fish samples. So both active and passive fishing techniques should be used to collect samples. Then the third is that sample all niches in the area, okay, including micro niches. It is expected that samples should be collected from all niches, including micro niches in the ecosystem. So, for instance, if we want to collect samples from this water body, it's not expected, it's not good to collect samples from just the middle of the river or to sample certain areas, maybe mud flats, and leave out some other very important micro niches. All niches should be sampled so that species that are found in such niches can also be collected. The fourth is that you should select definite sampling procedures which should last for many hours per trip. Definite sampling procedures should be selected. The trip may include night and day sampling because there are some fish species that are active at night, some are active during the day. Some species can easily be collected at night, while others are collected during the day. So both day and night should be sampled if the actual ichthyofauna composition of an area would need to would be represented. Samples should be collected both day and night. All niches should be sampled so that the proper representation of the biodiversity of the type fauna of the area should be recorded or known. Then the fifth uh, sampling procedure is that make regular sampling trips at least once or twice a month and should last over a period of 12 months to include at least one of each season in the location. Okay, regular sampling trips should be made once or twice monthly. Okay, should not be collect fish samples should not be collected just once in a month or once in a year or twice in a year or 6 months in a year. No, should be collected throughout the year to represent the sampling seasons. So to have a comprehensive data of the entire fauna of an area, the sampling should be done both during the dry season and the rainy season. For instance, the sampling should be done both during the dry season and the rainy season. The reason is that there are some fish species that that will be available during the rainy season that will not be available during the dry season. And there are some that are available during the dry season, but are not available during the rainy season. There are some fish species that are available only on specific months of the year. Okay, so if sampling is done in a way that it doesn't cover such months, such fish species will not be collected. So it's expected that the samples should be collected once or twice a month and should be done to represent the entire seasons. Yeah, the 12 months of the year should be sampled so that no fish species is left out in the entire final study to have a proper representation of the fish species in an area. It is very important that that's done. After obtaining the samples, you're expected to label the samples properly using serial numbers written with pencil of water-resistant ink on a waterproof material, such as a masking tape. So labeling is very important. So proper labeling of the samples should be done. Then basic data such as total length, standard length, fork length, and body weight can be measured in the field if time permits. So when after the fish have been collected, the fish should be measured. Basic data such as the total length of the fish. The total length basically is the measurement of the length of the fish from the snout to the tip of the tail. The fork length is from the tip of the snout to the inner part of the caudal fin, while the body weight is done by using a scale. All of this data should be measured. Even the girth of the fish can be measured in the field. Then the third is that you dissect each sample and preserve in 4% neutralized formalin. Okay, the fish should be dissected and preserved in formalin. Now I want to look at the precautions that should be taken in ichthyofauna studies. What precautions should you take when working on fish samples or when collecting fish samples and preserving them? The following precautions should be taken while engaging in ichthyofaunal studies. The first precaution is that field notes should be taken and should state the date, time, place, gear type or types, color, other observed behavior of fish, and other available data. So the field notes should be taken. Yes, it should be taken, and the date, the time, the place, the gear type or the types or color, and other observed behavior of fish and other available data should be stated in the field notebook so that it would basically assist in the explanation or in the reporting of the data that have been collected. Then don't allow formalin to get into contact with your skin because formalin can be irritating and could be carcinogenic. So it's good to avoid the contact, avoid contact of formalin with your skin. Then use hand gloves when necessary to avoid contact with formalin. Let us look at the gear types. We want to look at gear bias. Gear types used for sampling can be divided into two categories. We have the active and the passive gear types. Active gear includes those that are moved through the water either by machine or with human power. The active gears are moved through the water either by machine or with human power. You actively chase the fish with such gears, or the fish are actively being collected, while the passive gear is usually set and left stationary for a period of time. The passive gears are usually set and left stationary for a period of time. So that's the difference between the active gears and the passive gears. The active gear commonly used in inventory surveys include the seine nets, such as the beach seine nets and the pole seine nets. Then the trawl nets are also used, the deep nets, the electro stalkers or electrofishing, cast nets, and angling are all various active gear types. While the passive gears include the gill nets, the hook and line, the ammunition nets, and the minnow traps. So far, we have looked at various reasons or precautions to be taken while collecting fish samples and the various methods to be used. And in our subsequent videos, we'll look at detailed procedures for the collection, preservation, and identification of the ichthyofauna of an area. Please subscribe to our YouTube channel to watch more of this series on biological techniques.